Hi, this is Andrew Wolf. In this video, I'm going to talk about how mutations can occur during somatic cell division. And what is somatic cell division? Somatic cell division occurs when body cells divide to replenish themselves within a living organism. So that is when we've got a body cell here, and within the cell, we've got a nucleus with a chromosome, actually 23 pairs of chromosomes in it. And with somatic cell division, each of these chromosomes will directly copy each other. So they will create an exact copy of each other. And so we will end up with identical pairs with an identical copy of these same 23 pairs of chromosomes. Okay, so this is somatic cell division, or otherwise known as mitosis. So this is very different than what we see with sexual reproduction and meiosis, and this is sort of what I'm referring to here. Mitosis is, again, how our body cells replenish themselves on a day-to-day basis. So I'm very much simplifying things here, but during the process of mitosis, the body, the cell first has to replicate its DNA, and then it sort of has to replicate all of its organelles, and then it divides itself. Now the picture isn't very pretty here. These are supposed to be two identical cells, but you'll have to sort of use your imagination here. They have the same identical DNA, and they will end up looking like the same exact identical cells over time. Okay, and then when they want to replicate each other themselves again, they will again duplicate the DNA in the exact same fashion and divide themselves by mitosis, and we will end up with two more sets. Okay, and that's how the cells replenish themselves within the body, by cloning themselves. Now what's interesting is we've got about 50 trillion cells in our body, and of those 50 trillion cells, about 100 billion or so of those cells are replicating themselves every day. Okay, so that is how much turnover we have. And some cells turn over more rapidly than others. For instance, neurons and skeletal muscle cells and cardiac muscle cells are thought to never replicate themselves. But then we have cells like our cells in our hair follicles and cells in our gut mucosa that are very rapidly dividing and replicating themselves on a daily basis. Now, if we think about these numbers here, of our 100 billion cells that are replicating themselves every day, each cell has 20 to 25,000 genes within them. Okay? Now, each of these genes is generally copied exactly, right? And there are no mistakes. But there is an error rate in replication that is thought to be, and no one knows the precise number here, but it's estimated to be about 1 in 1 million to 1 in 10 million. So it sounds like a pretty rare error rate, right? We have a very low error rate, but the thing is, we're talking about massive numbers. So we've got 100 billion cells replicating each day with 20 to 25,000 genes being replicated. So we are actually talking about 2.5 quadrillion genes replicated in our body every day. So even with a very, very minuscule error rate, we are actually talking about tens of millions to hundreds of millions of genetic errors that our cells make every day. So, what does that mean? Well, you would think that after, decades of life, that we would all be that our cells would be mutated messes. So why are they not? Well, fortunately, most of the mutations are meaningless, right? So we could have mutations of some nucleotides, and that doesn't necessarily affect the proteins that are produced. So it doesn't have any meaningful effect on the process. Also, our cells have a whole bunch of protective mechanisms. And these are really important to understand cancer. So what are these protective mechanisms that protect our DNA? Well, we have proteins that we make that repair damaged DNA. We have proteins that actually detect damage to DNA and stop mitosis when there is significant damage. And then when things get really bad, we actually have proteins that initiate apoptosis when there is very severe mutations. Okay, and this is just a list of a few of the protective mechanisms, but this gives you an idea of how our cells, our bodies can have tens of millions to hundreds of millions of mistakes made in our in the copying of our DNA on a daily basis, and still have the majority of our cells be normal. Now, why am I talking about all this? Well, because cancer is cancer is a disease that is caused by acquired mutation. That is, mutations that are acquired primarily through somatic cell division. Now, that's not to say that there are some cancers that are inherited. There can be an inherited component. But it's interesting because the inherited component usually just sort of sets people up for some more acquired mutations that leads to the actual cancer. So what happens during mitosis that can cause mutations? Well, a few things can happen. We can have what's called a point mutation. Now, ordinarily during mitosis, we're going to have new DNA that's formed, and what happens then is we have we have our nucleotides match themselves up to their corresponding pair, right? But what happens is, if you remember my prior video, so this is what the normal sort of base pair matches would be. Now, what happens here is instead of... Okay, so I just had to go back and write in the typical, the base pairs that always occur between the nucleotides. So what would happen here is as the DNA is forming itself and getting ready for mitosis, replicating itself, with a point mutation, we would have, for instance, instead of a adenine connecting to a thymine here, it might accidentally, we might actually accidentally have a cytosine fill in here. So that is what would be called a point mutation. Or we could have, I'm going to add the base here, or we could have what's called a insertion, where we're going along here and we connect up our thymine, and then we add in an extra cytosine here, and then go on to put in our guanine. Okay, so this would be called, obviously, an insertion. Okay, now an insertion could be one nucleotide, could be 10 nucleotides, or it could be an entire gene. It could be hundreds. So it could be it could be a small insertion, it could be a large insertion. Same thing on the opposite side, we could have a deletion during replication here. So where we leave out one nucleotide. So that would be a deletion. And likewise, the deletion could be one nucleotide or many nucleotides. Okay? Now, These are generally relatively small, and generally, point mutation and small insertions and deletions are probably not going to be very meaningful. So they were sort of what I was talking about before. They're probably going to not have significant effects on the protein's structure and the protein function that the cells produce. What tends to be more problematic is when we start having issues at the chromosome level, where we have sort of whole pieces of arms of the chromosome that are moving from one chromosome over to another chromosome. And then you end up with, and these are called translocations. Then you end up with like gene splicing, where you have two genes that are getting put together that sort of create a new gene fusion, a new fusion gene that creates another protein that was not there before. So it codes for a whole new protein because we've sort of fused two genes together that weren't together before. So this gene from chromosome 1 fuses with a gene, comes and sits right next to a gene that was on chromosome 4. The two of them fuse together to create a whole new gene, and now that gene is coding for a new protein. Okay, so that kind of thing, when we're talking about whole chunks of a chromosome, that tends to be much, much more significant. So there's a very real example of this. For example, there's a gene down on chromosome number 22 that is called ABLE. I don't know why it's called ABLE, but that's what it's called. And there's a gene on chromosome number 9 that's called BCR. And during mitosis, there's sometimes an error where the BCR gets transposed and actually gets copied next to the ABLE on 22. And then we end up with a weird gene fusion called BCR-ABLE. And this creates a new protein, a tyrosine kinase protein, that is actually part of the pathophysiology of CML, or chronic myelogenous leukemia. Okay, so now that you understand how mutations can occur during somatic cell division, or mitosis, this really kind of sets you up to understand oncogenes and tumor suppressor genes. So in the next video, I'm going to talk specifically about oncogenes and tumor suppressor genes, how they can occur during mitosis, and why they are so important in understanding the development of cancer.